Hi, everyone. Okay, great. So I'm going to get started straight away because I'm slightly conscious of the time this evening. There's quite a lot I want to cover with you guys. So I hope you're all getting on okay. I'm sure you've had a degree of information overload at this stage. Hang in there. You've 1 week left and then there's a break for a week. And I definitely encourage you, if you can, next week to just take the week off and give yourselves a bit of space. As I've also mentioned lots of times before, the content that you've covered here, you're going to cover again. So you'll cover it in the next unit, in unit 3, so the cell signaling will come up again. And you'll cover antioxidants in unit 7 and again next year. You'll cover metabolism that we'll look at this evening in unit 8 and again next year, and in unit 10 actually. So all of this content you're going to see again and again. So as I said, please don't worry about trying to remember all of this time around. But what I am going to do this evening is go through, as we would do usually, the summary of the 3 PowerPoints and then answer any questions that have come up on the forum within them. There is quite a bit to cover this evening because there's a couple of key concepts that I really want to support you in getting a good foundation in. So we'll see how we go. And if we've time at the end, we will look at the assessment paper and the question. But if you have any questions again in relation to the assessment, you can ask me them at the end. And then also, if anything comes up during the week before Friday, you can ask me that as well. So let's begin. So the first PowerPoint that we will look at is metabolism. So we're going to look at micronutrients in metabolism and energy production. So this is really important, what we're going to cover, because it comes, so many of the clients that come to see you will be suffering with low energy. And the degree of the low energy to which they'll experience is on a big spectrum, where some people will just come in and feel a bit tired, whereas other people will come to see you. They may have something called chronic fatigue, or they may have quite severe fatigue. And at the center of fatigue is the energy production pathways. So it's quite important. Can everyone hear me actually just before I go on?
Loud and clear, Maura, yeah.
Okay, brilliant, thanks Dermot. Yeah. At the center, what we're going to talk through now, it's quite complex, and I know you hopefully have already gone through it already. But what I want to do is spend some time talking through it, and if you have any questions, ask me. And then, as I said, it's going to come up again in Unit 8 and Unit 10, so don't worry about remembering it all this time round. But we will go through it now in a good bit of detail. So the first thing to look at when we're talking about metabolism is there's 2 types of reactions. So we have catabolic reactions and anabolic reactions. And metabolism refers to all of the reactions that are going on in the body. So catabolic reactions refer to complex molecules being broken down into simpler molecules. And a lot of the time when we're talking about metabolism and energy production, that's the kind of reactions that we're talking about. And we're going to look at some of those pathways this evening. So this is a really important process where you have complex molecules, so glycogen, proteins, and triglycerides, being broken down into their simpler molecules, such as glucose, amino acids, glycerol, and fatty acids. The catabolic reactions tend to release energy, whereas anabolic reactions tend to require energy. And the energy currency of the body, so the money, or the euro or pound, whatever, wherever you are, is ATP. And that's where we store energy within the body. And so ATP, adenosine triphosphate, is a molecule which contains 3 phosphate bonds, or 3 phosphate molecules bonded together. And what happens is in the second and third phosphate molecule, the last bond, that's where a huge amount of energy is stored. And when you break that bond apart, you release energy and you form ADP, and you release the phosphate. There's a huge amount of energy being stored in this bond, and when you break that bond, energy is released. So what we're really concerned about as nutritional therapists is looking to see, well, how is that energy created, or how is ATP, let's say, taken from food, or how is energy taken from food and converted into ATP, the energy currency. And it's a really quite a long process to do that, and we'll look at that this evening. And that's a really important catabolic reaction. So actually glucose, amino acids, and fatty acids, these three here, and glycerol in a sense, they all can go a step further, and they actually can get broken down themselves into ATP, and that's what we're going to look at. So that's what catabolic and anabolic reactions are. And sometimes you'll see that in papers. You'll see the catabolism and anabolic reaction. So that's all they're referring to. The key thing is that catabolic reactions release energy, whereas anabolic reactions require energy, and that comes from ATP. Okay. So all the reactions in the body, and when we're talking about reactions, we're talking about molecules getting converted from one into the other, and this will make more sense when we look at specific reactions later on. But all of these reactions, for the most part, don't occur just on their own. They require these things called enzymes. And enzymes are really important because they catalyze, or they speed up the rate of reactions. And so how they work is basically they act as a site a lot of the time. So if you see here, if we start here, so here's an enzyme. And I know I've mentioned this quite a number of times, but enzymes are made from proteins. So that's a really important key or point to note. That enzymes are made from proteins, and the recipes for making the enzymes are found in your DNA. And a lot of the reactions in the body, or a huge amount of them, are dependent on enzymes. So this is an example of an enzyme. Sucrase is an enzyme. And what it does is it helps you to break down sucrose, so sugar. So we often call the compound that's getting broken down the substrate. So in this example, you have your enzyme, and which has this kind of active site. And a lot of the time, the active site is determined by the shape of the molecule. So you can see here that sucrase sits into the enzyme, and then it sits in, and a reaction actually occurs while it's sitting in the enzyme. So here, for example, sucrose is broken down into glucose, and then the glucose and fructose are released. So you can see here's our enzyme. Sucrose, which is our substrate, sits into it. It breaks in while it's sitting in the enzyme. The molecule gets broken down, and then it gets released. So the conversion of sucrose into fructose and glucose is dependent on the enzyme. And if you don't have the enzyme, the reaction may not occur. And that's why we're really concerned with enzymes, digestive enzymes. And again, you look at this in units 4 and then again in 7. But enzymes are released from, some of them, most of them are released from your pancreas, or some of them from the small intestinal lining into the small intestine. And that's where you break down your macronutrients, so your proteins and your fats and also your carbohydrates. And they're dependent on all these different enzymes. And I'm sure many of you are aware that you can buy digestive enzymes in a health food shop, and if for some reason people aren't producing or releasing them themselves. But this is how they work. So that's the first thing in terms of the basics of enzymes. Then what we have is something called cofactors and coenzymes. And this is quite important because it relates to, or it explains why micronutrients are so important in metabolic reactions within the body. So cofactors are a lot of the time inorganic, so they don't contain carbon, and they are required to increase the rate of a reaction or to catalyze it. And sometimes they'll sit into an enzyme, or sometimes they'll actually kind of act independently. They'll a lot of the time be a part of an enzyme, but they're a little bit different to a coenzyme in that they don't affect the shape of the enzyme. They kind of act slightly differently. And we'll look at that when we look at an example of the P450 enzymes. But the key thing is that cofactors are a lot of the time ions or minerals, and they have an important role in increasing the rate of a reaction. So sometimes people don't realize that there is a difference between cofactors and coenzymes. So that's something that's important to note. So here our cofactors are iron, copper, magnesium, and zinc. And again, I'll show you an example of how iron affects a chemical reaction in your liver. But you also then have coenzymes. So coenzymes, a lot of the time, they contain carbon. They're organic molecules. And a lot of the time, coenzymes are made from vitamins. So a lot of the time, B vitamins in particular, so B1, 2, 3, 6, biotin, and B12, act as coenzymes. And so how coenzymes work is they bind loosely to the enzyme at the active site, and they aid the enzyme activity. So they actually play a role in creating, in a sense, the active site. So they're really essential to the enzyme for it to function. So you'll see here, this is a good picture because it demonstrates how it works. So you see here's our enzyme, and here's the active site. And so these are our different compounds, but so this here is, if we take this example first. So here what we have is, here's the enzyme and here's the active site. And the coenzyme sits in here and it affects the structure of the active site. And you can see here, this part of the coenzyme is a vitamin. So coenzymes can be actually quite big molecules. Sometimes they're just a vitamin, and sometimes they're a number of different molecules, and we'll see an example of that in a second. But here you can see that this vitamin plays a really important role in creating the shape that forms the active site. So this molecule here, CD, sits into the active site, the reaction occurs, and C and D break apart. And this is our product. In the same enzyme, you can have a different type of coenzyme, so it has a different shape. And you can see here A and B sit in, they create a product, and then they're released. So the same enzyme can have one enzyme can have one active site, but depending on the type of coenzyme that sits into it, it can actually just determine what reaction occurs. So you can see here, enzymes or coenzymes, I should say, are really, really important, and specifically vitamins, for enzymes to function. And sometimes they're really big, complex molecules. So this is an example. So pyruvate dehydrogenase is an example of a multi-enzyme complex. So we'll look at this in a second. It plays an important role in the energy production, in how you break glucose down into energy. And don't worry about these terms for a second. But pyruvate dehydrogenase, it actually requires 5 coenzymes and 1 metal ion factor. So it requires B1, lipoic acid, which is a coenzyme, lipoic acid, which we'll look at later on, it's a compound, B2, B3, B5, and magnesium. So it's a really, really complex enzyme that requires a number of coenzymes and a cofactor. And so all of these nutrients are really important for energy production, because if you, for example, were deficient in B1 or you were really low in magnesium, it may mean that you don't have enough magnesium to ensure that this enzyme is functioning properly, and therefore you can't break down energy or glucose into energy, which we'll look at. So the production of energy in particular is really, really dependent on magnesium, B vitamins. We'll also look at other things later on. But one pathway in particular requires this enzyme complex that is highly dependent on these 5 coenzymes and a metal ion cofactor, magnesium. So energy production, it's a highly micronutrient dependent process. So that's the first thing to look at. So as I said, these are if we come back just here for 1 second before we go on, enzymes are essential for metabolic reactions to occur. And they require sometimes cofactors, which tend to be metal ions, so iron, copper, magnesium, or zinc. Or they can rely on coenzymes, which a lot of the time are vitamins, which bind at the active site. And B vitamins are often coenzymes. And you can see here they're highly dependent. The enzyme is highly dependent on the coenzyme. A lot of the time it's a vitamin. And an example here is sometimes you might have a really big enzyme complex that requires not just coenzymes, but also a cofactor. So here, this guy requires 5 coenzymes and 1 metal ion cofactor. So it's highly micronutrient dependent. And the conversion of one compound, it is a compound, the conversion of one compound into another is highly dependent on enzymes. So if you're low in these micronutrients, the chemical reaction might not occur. And we see this a lot of the time with people who have energy issues, fatigue and chronic fatigue syndrome. So if we look here, this is in this PowerPoint, I know towards the end it goes into a little bit of what happens with amino acids and also what happens with fats. And I have left that out because I don't want to overload you this evening and there's not enough time. And you'll come back to this again in Unit 8. You'll have covered it yourselves. You'll also come back to it in Unit 8 and then again in Unit 10. But what it will be good to focus on this evening is looking at how glucose in particular is broken down into energy. We'll cover lipids and proteins, but we'll focus mainly on glucose. So when you eat carbohydrates, no matter what form of a carbohydrate you eat, whether it's starch, disaccharides, all of them end up getting converted into glucose eventually in the digestive tract or in the liver. And glucose here goes through a number of steps in order to produce energy. So it goes through this process called glycolysis. It goes through something called the link reaction, and there's something called the Krebs cycle and the electron transport chain, which we're going to look at. Proteins can get converted into amino acids, which it can also feed into this cycle, and fats get broken down into fatty acids and glycerol, which also feed into this cycle. But what we're going to look at here is first glycolysis, then the link reaction, and then the other steps as well. So this is kind of I just want to pull up this one. So this is the 4 steps or the 4 stages. So the first one is when the oxidation of glucose means the breaking down of glucose to produce ATP. So remember, ATP is the energy currency of the body. All the energy that's in glucose gets stored or released and put into ATP. And then ATP can travel around the cell for the most part of the body and be used for anabolic reactions which require energy. So there are 4 steps in this process from going from glucose to ATP. So the first one is glycolysis, then you have something called the link reaction, then the Krebs cycle, and then the electron transport chain. And it's quite a complex process. And this diagram basically shows it here. So this is the first step. If we go, you eat food, gets broken down into glucose. And so first of all, glucose can get converted into pyruvic acid through glycolysis. Then a reaction occurs called the link reaction, where you convert pyruvic acid into acetyl coenzyme A. Then acetyl coenzyme A, it moves from the cytosol, which is the liquid in the cell, and it moves into the mitochondria. And in the mitochondria, acetyl-CoA gets converted gets fed into something called the Krebs cycle, which we'll look at. And then it releases it releases the electrons and stores them in something called NAD. NAD it acts as an electron carrier. And NAD then moves to something called the electron transport chain. And in the electron transport chain, energy is converted into ATP. And so the structure of the mitochondria is really, really important. You can see here it's a double membrane structure. So you have this outer membrane here, and then you have this inner membrane that kind of has these folds called cristae. Okay. And so two really, I'm going to kind of keep going on, say this a few times so it'll kind of hopefully go in eventually. But the key thing, the two really important things that occur in the mitochondria is the Krebs cycle and the generation of ATP at the electron transport chain. And the electron transport chain, it sits on this inner mitochondrial membrane, and it's really dependent on the structure, or it is the structure, but the structure, the intactness, that's the word, the intactness of the membrane. So remember, we looked at the membrane last week and the week before. Your membranes, the cell membranes in the body, whether it's the big cell, the cell itself, the big cell membrane, or the mitochondrial membranes, they're made up of this bilayer of phospholipids. So you've got two layers of phospholipids, and a chunk of that is made up of fatty acids, which kind of form this, in a sense of kind of a fatty barrier in it. Okay, so the intactness of the phospholipids in the cell membrane is really important for your electron transport chain to function properly. And if it doesn't function properly, you have issues with energy production. And this is what happens eventually. What some people might develop is something called mitochondrial dysfunction, and it can play a really important role in chronic fatigue syndrome, for example. So the intactness of this membrane is really important. And we'll look at what can damage that. We'll touch on it in the next PowerPoint, but you'll also come back to it again, as I said, when you look at energy imbalances in Unit 8. So that's kind of an overview of the different steps. You can see them here. And this is, again, remember, glucose, it's getting converted into something called pyruvic acid, but glycolysis is this step. Pyruvic acid gets converted into acetyl-CoA. This is called a link reaction. Acetyl-CoA feeds into the Krebs cycle and generates the production of NAD, which is where we're storing all our electrons, which are carrying the potential energy. The electrons are fed into the electron transport chain, and in that process you make ATP. Okay, and so now hopefully you see why the mitochondria are called the energy houses of the cell, because that's where a huge part of energy production occurs. This is another overview. So I'm going to talk you through this. Hopefully you'll see it from different perspectives. I'm just going to show you different parts, and hopefully it'll start to make sense. I know hopefully you've already covered this, but I just want to kind of reiterate the key points. So this is another overview, and you can see the different amounts of ATP getting made. So here's the 4 steps. You've got glycolysis, the formation of acetyl-CoA, or sometimes it's called the link reaction, the Krebs cycle, and the electron transport chain. And this process is called, a lot of the time, cellular respiration, or they call it the oxidation of glucose. Okay, so let's have a look at it this way. So glucose gets converted into pyruvic acid, and this process generates 2 molecules of ATP. And when you think about it, in total, I think you make about 36 molecules of ATP in total. So through breaking down 1 molecule of glucose. So the fact that in this step you just make 2, it's not that much energy when you think of 36 as the whole, all the ATP you could potentially make from glucose. So this step here, this is sometimes called as well anaerobic respiration, because you can convert glucose into pyruvic acid in the presence of no oxygen, but that process only generates 2 molecules of ATP. Okay. So once glucose is converted into pyruvic acid, it can then get converted into something called acetyl coenzyme A. And this process generates 2 molecules of carbon dioxide and 2 molecules of NAD. So don't worry about remembering. You don't need to remember how many molecules of ATP or NAD are generated at each stage. I'm just giving you some detail here, but all you'll kind of hopefully take away is there are these 4 steps, and 2 of the major parts occur in the mitochondria, and a large part of energy is actually made in this last stage, which I'm going to show you. So we've got glucose converted into pyruvic acid, 2 molecules of ATP and 2 molecules of NAD. For the moment, just think of NAD as an electron carrier. And why that's important will make a bit more sense when you look at the electron transport chain. But just think that NAD is a molecule. It's actually derived from vitamin B3, but NAD carries electrons, okay? And that's really important because electrons help to generate energy later on. So glycolysis generates 2 molecules of ATP, 2 molecules of NAD. Then we've got pyruvic acid getting converted into acetyl-CoA. That generates another 2 molecules of NAD and 2 molecules of carbon dioxide. And then acetyl-CoA feeds into the Krebs cycle. And the Krebs cycle is a really, really important step for a number of reasons. It generates some ATP, 2 molecules, but what it really does is it generates 6 molecules of NAD and 2 molecules of FAD. So FAD is also an electron carrying molecule. Think of it like a postman having a bag and there's loads of electrons in it. So NAD and FAD both have this capacity to carry electrons, and that is really important for later on. So this is where a large proportion of these electron carrying molecules are generated. So that's why the Krebs cycle is so important, that you carry or you generate NAD and FAD in this step. And this is a cyclical set of reactions. We'll look at the reactions in a second, but it just keeps going round and round. But hopefully acetyl-CoA keeps feeding in. So after these molecules of NAD and FAD are generated or take on the electrons, they move to the electron transport chain, which is sitting on the inner mitochondrial membrane. And these electrons, they feed through a chain of protein carriers. The proteins sit on the membrane. And the electrons move through the protein carriers. And in the presence of oxygen is really important here, they generate ATP, or they produce ATP from ADP and phosphate. So what happens is ADP and phosphate are bind together, and your energy from your electrons is stored in ATP. But in order for this step to occur, you need oxygen. And if you don't have oxygen, this won't work. And that's why energy production, cellular respiration is dependent on oxygen. And so that is kind of a key part, is that oxygen is needed for this electron transport chain to work. What's also needed for the electron transport chain to work is an intact cell mitochondrial membrane and a set of functioning integral proteins, which we're going to look at. Okay. So that's an overview of glucose metabolism or cellular respiration. What I'm going to show you now is the different micronutrients that are required at this stage, and this stage. But before we do this, I just kind of want to summarize by showing you that a little bit of ATP is made in the first step, a little bit in the Krebs cycle, but the majority is made in the electron transport chain step, and that requires oxygen. And carbon dioxide is produced as a byproduct or a waste that has to be removed. So energy production or glucose metabolism requires oxygen to work and produces carbon dioxide as a result, which then has to be removed from the body. So that is glucose metabolism. And now let's look at the different micronutrients that are involved. Okay. So in the link reaction, so remember the link reaction is where you convert pyruvate into acetyl-CoA. It requires the enzyme that we looked at earlier called pyruvate dehydrogenase complex. And pyruvate dehydrogenase complex, it basically enables the conversion of pyruvate into acetyl-CoA. And it has a number of coenzymes. It has B1, B2, B3, B5, and lipoic acid. And it also has a cofactor, magnesium. So this is one of your assessment questions. It's question 4. So which of the vitamins, or what are the cofactors and coenzymes for the link reaction? So this is the link reaction, and these are the coenzymes, or these are the coenzymes, and this is the cofactor. And so this is kind of the detail of what is occurring here. You don't need to worry about too much. The key thing just to take from it is that pyruvate gets converted into acetyl-CoA. In order for this to happen, you need pyruvate dehydrogenase enzyme. And this enzyme is dependent on 5 coenzymes and 1 cofactor, namely B1, 2, 3, 5, lipoic acid, and magnesium. So if you're low in these nutrients, this enzyme may not function properly. And as a result, you may not be able to make acetyl-CoA, and as a result, energy. So hopefully you're starting to see how the enzymes are really important for the processes, or for the chemical reactions, and how then the enzymes themselves are dependent on cofactors and coenzymes. So if you don't have them, the enzymes won't work properly, and therefore the reactions won't occur. So that is the link reaction, and now we're going to look at the Krebs cycle. So again, you don't need to remember all of this, but it's just to show you the different nutrients that are involved. So acetyl-CoA comes in, and in the cytosol of the mitochondria, it feeds into this cycle it's sometimes called the citric acid cycle. Sometimes you'll see it being called the Krebs cycle. There's a few different names for it. All you want to kind of take from it is that acetyl-CoA feeds into this cycle, and it gets converted into lots of different compounds. Citrate is one of them. Malic acid is another. And all of these yellow carbons refer to different compounds. But all of these compounds get converted into each other, and in the process, you can release an electron and you add it to NAD or to FAD. Okay. So these chemical reactions release electrons that can get stored in NAD and FAD, which then move to the electron transport chain. But each one of these steps requires an enzyme. And so again, you can look this up, you can see all the different enzymes that it requires. You don't need to know that. What we're just trying to demonstrate to you is that step 2 requires iron, step 3 requires magnesium and B3, step 4 step 3 here requires B1, 2, 5 and lipoic acid, and 5 magnesium. So all of these micronutrients are dependent, or all these micronutrients are essential for the Krebs cycle to function properly. And so if you're low in B2 or you're low in B5 or B3, magnesium, iron, the Krebs cycle may not function properly. And as a result, you won't generate NAD and FAD, and you won't be able to power the electron transport chain to make ATP. So again, it's really important. If we just come back here, hopefully you're still with me. I know this is quite a lot to take in, but again, as I said, you're going to come back to this again and again. But it is good to look at energy production at this level, because you get to then see how important micronutrients are. So what we've just looked at here is the link reaction, the conversion of pyruvate into acetyl-CoA, which requires pyruvate dehydrogenase, and B1, 2, 3, 5 lipoic acid and magnesium. And then we can see the Krebs cycle here, This occurs in the cytosol of the mitochondria. It's dependent on B1, 2, 3, 5, I think, magnesium, iron, lots of different nutrients as well. And that produces NAD and FAD, which are carrying electrons. And then these molecules move to the electron transport chain. So if we have a look here, so this is the electron transport chain. I've got two different pictures, I think of, yeah. So this is the electron transport chain. It sits on the inner mitochondrial membrane. So if I jump back again once more. So this is our mitochondria. It's got an outer mitochondrial membrane, and then you have this inner mitochondrial membrane on the inside. And you can see here the electron transport chain sits on this. Now there are hundreds, I think thousands of electron transport chains in one mitochondria. They're all sitting along them or embedded within them. And so what are they made up of? So they are made up of, the electron transport chain, it's made up of integral proteins. So you can see here is the inner mitochondrial membrane. We've got our little phospholipids here, our bilayer. And embedded within the bilayer is integral proteins. So again, we looked at this in last week. We looked at the plasma membrane and how things move through it. And so these are the proteins. You've got 1, 2, 3, and 4. These proteins sit within the membrane. This is also CoQ10. And they do something really important. They allow protons to move from this inner mitochondrial space to the intermembrane space. So the intermembrane space is the space between the inner mitochondrial membrane and the outer mitochondrial membrane. It's the space in between. And what happens is NAD and FAD come along, and NAD and FAD they release electrons from the bond between NAD and H, NAD and H. And the electrons here move through the membrane. This kind of yellow line, these guys symbolizes electrons. So remember, when you have a H plus bonded to another atom, there is going to be two electrons, one from the hydrogen and one from whatever it's bonded to. And if you have a H plus, it's lost its electrons. So what happens here is the bond between NAD and H gets broken. The electrons are released. They move through the chain, and the H plus that's left moves from this space into this one. Okay. So again, you don't need to know this level of detail in terms of the bonding and the electrons, but sometimes for some people it makes more sense if we go into this detail. So just if I'm going to say this again, just once more, just hopefully if people haven't got it, it makes a bit more sense. There's going to be, in NAD, there is a bond between NAD and a H. Okay. And there are two electrons in that bond. And if they get released, a H plus ion is also released. Okay. So you end up, so this NAD is equal to NAD plus a H and two electrons. Okay. So when you release the two electrons, you're left with the H plus. So hopefully that's making sense. So the electrons move through the proteins within the membrane, and their movement actually generates an energy that allows this proton here to move from this side of the membrane to the other. And this is really important because, and hopefully you might remember this from last week again, there's more protons in your mitochondria. There are more protons in the intermembrane space. So when the protons move from here to here, they're moving against a concentration gradient. So remember, you have a concentration gradient when you have a higher concentration of something on one side of the membrane than the other. So in this example here, there are more protons in this inner membrane space. So when the protons are moving from this side to this side of the membrane, they are moving against the concentration gradient. And whenever you're moving against a concentration gradient, you need energy. And so the movement of these electrons through the integral proteins generates energy. It's a form of kinetic energy, basically. We don't need to go into, we definitely don't need to go into this, but it's just the movement of electrons is a form of kinetic energy that activates these carriers and pumps these protons from this side to this side. And so what happens here is you get a buildup of these protons in the intermembrane space. And if we move over here now, sitting within the membrane is also an enzyme called ATP synthase. And what that enzyme does is it synthesizes ATP. So with enzymes, you'll know if something's an enzyme if there's an A's at the end of it. And most of the time, the name of the enzyme kind of tells you what the enzyme does. So this is a really good example of that. This enzyme makes ATP, so it's called ATP synthase. And so this is, so I'll show you this here. So we have here, this is our ATP synthase. And it makes ATP from ADP, which is, it has 2 phosphate molecules in it. So it basically makes ATP from 1 phosphate molecule attaching to ADP. Okay, and it's in this bond here. So this is the ATP here. You've got 1, 2, 3 phosphates, and it's in this last bond here that all the energy, it's kind of amazing, all the energy that's in glucose gets put into this little bond here. Okay. And then this ATP can move off to different parts of the cell, and it can use the energy in that bond to create larger molecules. It can fuel anabolic reactions. So it can make other enzymes, it can make other proteins from amino acids. Any reaction where you make a bigger molecule from a smaller molecule, for the most part, you'll need ATP, and you'll need the energy that's in here. And so again, how is this energy converted? It comes again to this form of kinetic energy movement. And so what happens is the movement, these protons build up in this intermembrane space. And when they move from this side of the membrane to the other through this enzyme, they generate energy through kinetic energy. So again, kinetic energy is energy that results from movement. And so these protons, they move from this side to this side. As a result, they end up turning on the enzyme. I suppose a way to think about it is you have kind of those kind of, I don't know, in rivers, you have those kind of cogs where the water pushes I'm not giving you a good example, but it pushes this kind of wheel and movement occurs. And that's basically what's happening. Well, that's not basically what's happening here. But what is happening here is that the movement of these protons from one side to the other through this enzyme activates this enzyme and basically enables you to create ATP. So the energy moving from here to here gets converted into this. And then this ATP can go off and do what it needs to do. So that is the electron transport chain. As I said, it's a really important, a hugely important process in energy production. And hopefully what you're seeing here, how important are a number of different things. One is that the mitochondrial membrane is intact, because what happens is if the mitochondrial membrane is damaged, or there are basically, let's say, holes in it, sometimes you'll get a membrane that gets damaged and there might be some holes. The protons, they don't move down through the enzyme and activate it. Instead, they actually just move through here. And as a result, that movement can't be used to generate energy. So the intactness of the inner mitochondrial membrane is really important. The electron transport chain itself is really important. So these proteins, they are determined, we're going to look at in a second, they require certain micronutrients to function. So the electron transport chain itself is dependent on a number of nutrients. So you can see here, it's dependent on B2, iron, sulfur, copper, and CoQ10. And also actually B3. You can't see that here, but it is required. It requires B3. This diagram might show this a bit better. So this is the inner mitochondrial membrane here. These are the electron transport chain. And these are made up of proteins, and attached to these proteins, so here you have B2, here you have sulfur and iron, and this is CoQ10. B3 is in there somewhere as well. But so all of these, the chain itself is dependent on B2, B3, iron, copper, sulfur, and CoQ10. So again, if you're low in these nutrients, it might affect the chain. What I also want to show you is just one more thing, and it affects, or it relates to the next PowerPoint. So this process is dependent on electrons moving on their own through a set of proteins embedded in the membrane, which is quite volatile in a sense. Because remember, we looked at free radicals. Free radicals are unpaired electrons, and they're incredibly reactive and damaging, and potentially damaging. So the energy production process itself actually generates reactive oxygen species. So most of the time, or the majority of the time, what should happen is the electrons move through the chain and they bond with oxygen at the end to form H2O. So you can see here, H plus ions, electrons, oxygen form H2O. But sometimes what happens is these electrons leak. They might damage the membrane itself a little bit. Hopefully it gets caught by CoQ10, which is here. Or they kind of leak and they just attach to an oxygen molecule without a hydrogen, and they create something called a superoxide anion, which is a really reactive oxygen species. So energy production itself actually generates reactive oxygen species. And that's why antioxidants, again, we're not going to get to cover this in a lot of detail, antioxidants specifically within the electron transport chain, but you will look at it in Unit 8, you'll touch on it, and then again, you'll look at it in a lot of detail next year when you're looking at how to support people with energy production issues. But you have a certain antioxidant enzymes in this area that are there to kind of mop up any reactive oxygen species that are generated from energy production. So I think it's SOD, superoxide dismutase is a really important one, and glutathione peroxidase as well. These are really important antioxidant enzymes that negate the side effect of reactive oxygen species being produced by energy production. And this is one, energy production itself is one of the biggest sources of generating reactive oxygen species in the body. So just before we go on, I'm just going to give you just one more overview of this, because again, you're going to learn lots of different things within the course, and of course you're not going to remember everything. However, energy production, specifically cellular respiration, it is an important process over the next 2 years to become quite familiar with, because people are going to come to see you with energy production issues, and therefore it's good to understand the process by which they make energy and the different nutrients that are required. But as I said, this is a lot to obviously take in one module. You'll come back to it again and again. So glucose, and actually just on this, you'll see here, glucose is essential in cellular respiration, but amino acids can also feed in and create pyruvate, which then feed into acetyl-CoA. Amino acids can also convert to acetyl-CoA. And fatty acids and glycerol, which make triglycerides, they can also feed into acetyl-CoA as well. So all of all of your macronutrients can feed into the energy production pathway. Okay, so this is our overview. So again, you have 4 steps that are involved. So you've got glycolysis, the link reaction or acetyl-CoA, the Krebs cycle, and the electron transport chain. So there's 4 stages. In total, you make about 36 molecules of ATP. Remember, ATP is the energy currency of the body. But the majority of that ATP is made in the last stage in the electron transport chain, and that requires oxygen. You make a little bit of ATP in the first step in glycolysis, and this is where we make 2 molecules in the absence of oxygen. You make a little bit in the Krebs cycle, but the key thing in the Krebs cycle is that you generate or you convert or allow electrons to move from these compounds to NAD and FAD, where they can then move the electron transport chain and feed into the chain and make ATP. And the energy production pathway itself naturally produces waste, and one of the waste products is carbon dioxide. So that's carbon dioxide then has to leave the cell and get removed in respiration. All of these steps are dependent on micronutrients. So the enzyme that converts pyruvic acid into acetyl-CoA is pyruvate dehydrogenase. It requires B1, 2, 3, 5 lipoic acid and magnesium. The Krebs cycle requires a lot of B vitamins. It also requires magnesium and iron and CoQ10. And then the electron transport chain requires B2, B3, iron, copper, sulfur, and CoQ10. So most of the stages within the cellular respiration are dependent on micronutrients to function. And if you don't have enough micronutrients, you won't be able to make energy from glucose. And again, we'll come back to this again and again when you look at what goes wrong in energy production, but this electron transport chain is so important, and it's within the mitochondria, and you really need to have an intact membrane and also antioxidants to mop up the reactive oxygen species that get generated naturally in the process. So that is energy production. It does relate to the assessment questions. There's a couple of questions. This question 4 is listing the cofactors and vitamins, the coenzymes that are involved in the link reaction and electron transport chain. And then there's also question 3 asks you to describe the role of a coenzyme. So remember, it's a coenzyme, not a cofactor, and what they're derived from. So that's a key thing that people sometimes miss, is it's important to name what it's a coenzyme, not a cofactor. And do you need to learn about fermentation? You'll naturally learn about fermentation when you look at the digestive tract and microbiota and different things that happen. But in regards to this unit, no. So just before I move on and we go into looking at antioxidants, does anyone have any questions on this content? No, but a quiet group. Is anyone lost and doesn't understand it? Any brave soul want to admit it?
Not lost, Maura, but I have a question, if you don't mind. Why is it, I mean, clearly it's essential that we have the cofactors and the coenzymes, but surely it's equally essential that we have the enzymes themselves as well, right?
Yeah, 100%. That's
why is it that we always hear people being told, oh, you have to take vitamin supplements, you have to take but you rarely hear about people being told to take enzyme supplements?
So that's a really good point. So You can take digestive enzymes, but you can't actually take enzymes in your body. So you have to make the enzymes from in your ribosomes. And so this is really, what you're bringing up is a really good point. And what can happen with people is people who have metabolic issues, they have SNPs in their enzyme. It some SNPs in the genes that code for the enzymes. And as a result, they might have, let's say, there's people with chronic fatigue who don't have enough enzymes, these guys, to support the Krebs cycle. Or there's research to show now that they're not able to convert glucose into pyruvate effectively because they're missing some of the enzymes as well. So that is what happens. But the problem is that you can't give those people those enzymes. And you can sometimes give them extra micronutrients that might enhance what they already have, but that you can't take the enzymes. Okay. But that's, again, Dermot, that's a great point, because that's where a lot of people's issues lie. And when you learn about SNPs, you learn about SNPs as well, and detoxification enzymes. And that's kind of where you have genetic abnormalities that affect people's energy.
Okay, thanks.
Okay. Okay, brilliant. So Anne-Marie, I was lost around the electron transport chain, but hopefully it makes more sense. Okay. For anyone who is kind of lost and that, you know, they've kind of gotten, let's say they were able to follow up until this step or these, and they don't really understand exactly what's happening at the chain, don't worry. For the moment, just take from this that you need these micronutrients as cofactors, and kind of for the assessment, definitely, you've obviously got to put in what they are. And hopefully then when you come around to this again in Unit 8, and you've covered a bit more content and you've heard of these themes again and again, this will come more to life. I would say that when you do come around to Unit 8, if you're still finding it difficult to understand this, ask loads of questions in the webinar, and for the in-class students, ask loads of questions, because sometimes it's just a small little thing that blocks someone from getting it, and then when they ask a question, it clarifies, it creates clarity and they get it. So again, don't worry if you don't get it now, but definitely come Unit 8, if you're still really confused, ask then the unit leader. Okay. So that is the energy production. Now we will look at antioxidants. Okay. So again, this PowerPoint also feeds into your assessment. So one of the questions is, there's a couple of questions. Well, there's a three-part to the question. So it's list the micronutrient cofactors for the antioxidant enzymes, explain a little bit about the antioxidant network, and also which well-known group of phytonutrients is known to exhibit antioxidant and pro-antioxidant effects. That is the question that relates to this, and hopefully through this PowerPoint that will all become clear. Okay, so let's first look at free radicals. So free radicals are chemical species with an unpaired electron in the outermost shell. They are produced when a covalent bond is broken and one electron from each pair remains with each atom. Having an unpaired electron makes a free radical unstable, highly reactive, and destructive to nearby molecules. This is a good time to go back to part 2 and the basics of biochemistry. So remember, in part 2 we looked at the structure of an atom. So all atoms have this kind of uniform structure where they have protons and neutrons in the center, in the nucleus. The protons have a positive charge, the neutrons have a neutral charge, and then these electrons move around these shells. So electrons have a negative charge and they spin around these things called orbits. In reality, they don't move around in this lovely unified way. They're a little bit more disorganized in that. And they do move definitely around in circles, but they're kind of a bit more flexible in where exactly they're moving. But for chemists' purposes, they kind of put them into these shells. So the first shell carries 2 electrons, and then the second shell carries 8. And so electrons will always, or not electrons, atoms will always want to become stable. So most atoms, so for example, carbon is an atom, oxygen is oxygen is an element. One oxygen is an oxygen atom. Hydrogen is an element. One hydrogen is one hydrogen atom. And so most of the time, elements don't exist as just atoms floating around in their own. They're usually bonded to different things. And what drives the bonding is this need for atoms to become stable. And what makes them stable is having 8 electrons in their outermost shell. Unless they're helium and hydrogen, and they have 2 in their outermost shell. But for the most part, we want 8 electrons in the outermost shell. So for example, oxygen has an atomic number of 6. It has 6 protons and 6 electrons. It has 2 electrons in the first shell, and then it has 6 electrons in the second shell. So it wants 2 electrons to become stable. And most of the time, what it will do, it will share 2 electrons with another oxygen atom or another atom. It forms covalent bonds. Another example then would be, let's say, carbon. So carbon has an atomic number of, I should have said oxygen has an atomic number of 8. It has 2 electrons in its center shell and 6 electrons in its outer shell. Carbon has an atomic number of 6. So it has 2 electrons in its first shell, and it has 4 electrons in its second shell. So carbon needs 4 electrons to become stable. And it does that by, a lot of the time, bonding covalently to either 3 other or 4 other carbon atoms, or it might bond to 4 hydrogens. It'll form 4 bonds. If it shares 2 electrons with another carbon, it's called a double bond. But don't worry about that again for the moment. Just take for this that you want 8 electrons in the outermost shell. So this is, now these guys have more. You can have more, but just ignore that. So this here is a free radical. It has an unpaired electron. So electrons, they don't just also want to have 8 electrons in their outermost shell, they also want to have paired electrons. So it's really reactive because it has 1 electron on its own. So what happens is it takes an electron from an antioxidant and it becomes stable. So that is kind of what free radicals are. They are either atoms or compounds where you have an unpaired electron, that as a result makes it quite reactive. And so why we care about this is that free radicals can create damage within the body. They can damage tissues because they're so reactive. And so what we're going to look here at, Patricia sent in a question. So she said, in page 11, the PowerPoint says that the biggest source of free radicals is the metabolic processes of our bodies, which in itself is quite shocking. So you've seen there, Patricia, that ATP production in itself naturally generates free radicals. So it's actually a highly oxidative generating process, oxidative stress generating process. So she says then proceeds to name a few, which most of them I understand, but I would really like to know what oxidation happens in the endoplasmic reticulum. What is the role of cytochrome P450? So I'm going to explain this to you. This will come up again in Unit 7, but I'll give you kind of a taster of it now. And then also how metal ion cofactors undergo autooxidation, and what carrier protein are they bound to in order to prevent autooxidation. So I'll explain to you a little bit more about what autooxidation is and how ion cofactors in P450 work. So I'll give you an example. It's way beyond the scope of the course to go into this in detail. And if you could also please talk through what is happening with the lysosome in relation to the enzymes named in the little box pictured on page 11. I couldn't find any information that would make sense to me. Could it be related to an immune response? And the answer to that is yes and no. So we'll go through this now, Patricia. I'll show you. This is the picture here. I'll talk you through a little bit about what happens here, what happens in these sites, and here as well. So we've looked. So reactive oxygen species again are species that have a free radical attached to them, and they have the potential to damage unsaturated fatty acid, DNA. They can generate glycolated products. They basically can damage the body. And there are sources of free radicals within the environment. So smoking generates free radicals. Certain pesticides can generate free radicals. Heavy metals, so mercury itself naturally generates reactive oxygen species. And there's a number of other different things, like UV light generates reactive oxygen species. So there's a lot of external things that generate reactive oxygen species. But as we've also noted, they're also generated within the body itself as well. And so one of the biggest sources of reactive oxygen species is the mitochondria. So we just looked there at the electron transport chain, and you can see that when the electrons move through the chain, most of the time they should bond to oxygen at the end and hydrogen and create water. But sometimes they leak, and they might actually damage the mitochondrial membrane if there's not enough CoQ10 present. Or they leak and they generate something called a superoxide anion and hydrogen peroxide. And these themselves are free radical or reactive oxygen species. And if you don't have enough antioxidant enzymes to kind of basically break them apart, you'll have reactive oxygen species and oxidative stress. So the antioxidant enzymes within the mitochondria are really, really important to negate the effects of the byproduct of the energy production process itself. Peroxisomes are organelles within your cells. They contain antioxidant enzymes, or oxidative enzymes, I should say, and hydrogen peroxide. Hydrogen peroxide itself is a reactive oxygen species. So it basically is reactive, and it can damage things. And it's actually a process that's used to break down debris within the body. We actually use hydrogen peroxide to break down old worn out compounds, so that they can then be removed or degraded. So kind of clearing out waste from the body in itself generates reactive oxygen species, but hopefully it's contained within the peroxisomes. But what you also have here is, if we take this guy for an example, so phagocytes and lysosomes. Phagocytes are immune cells, and when you come to the immune system, in Unit 5 after Christmas, you learn much more about phagocytes. But basically, phagocytes are immune cells that they're part of the innate immune system, the first line of defense. And what they do is they engulf compounds that they see on cells that are foreign. So they have the capacity to take in microbes and damaged cells that have been infected with viruses, and also compounds, and break them down. So they have, remember last week we talked about the methods of transport, and one of the methods was endo and exocytosis. So endocytosis is where the cell kind of takes something from the outside and brings it into itself, and that's what phagocytes do. So they engulf a cell or a compound. And how they then break down the cell is they use superoxide anion or different reactive oxygen species to actually break it down, and lysosomes do the same. And how these guys generate the superoxide anion or the reactive oxygen species within the cell is through these enzymes. So one of them is NADP, which I'll show you. NADP oxidase is an enzyme that has NADP, and what happens is it converts oxygen into this superoxide anion. So oxygen gets converted into the superoxide anion. You're left with NAD+. Don't worry about that for the moment. All you need to know is that this enzyme, NADP oxidase, generates this superoxide anion, which then can go and break down things that are necessary. So reactive oxygen species aren't all bad. We need them for our cells, or particularly our immune system, to function. It's when you have an overreaction, too much of it, or not enough antioxidant enzymes to negate the effects of it, that's when you get kind of problems. So that is the enzyme NADP. Oxygen gets converted into superoxide anion, and then it can go off and break down what it needs to in the cell. So whether it's the phagocyte or the lysosome, which is it's an organelle in your cytoplasm, it's in the cell itself. That's those cells. Then you have, within the endoplasmic reticulum, you have these enzymes called cytochrome P450 enzymes. And so these guys, again, we're kind of jumping ahead here. But these enzymes, they are the CYP450 enzymes. They are membrane-bound enzymes. They sit on the cell membrane. And in this case, they sit on the membrane of the mitochondria or the endoplasmic reticulum. So remember, the endoplasmic reticulum is an organelle within your cell. And so they have a really important role in biotransforming toxins and basically playing a role in detoxification. And so you're going to learn a lot about this in Unit 7. You'll learn lots of detail about how the phase 1 enzymes work. But what they do is they actually generate free radicals. So I'll show you here. So your phase 1, your CYP450 enzyme is part of phase 1 of detoxification. Okay, and these enzymes, they're membrane bound. They sit on the membrane, whether it's the mitochondria or the endoplasmic reticulum. And at the center of these enzymes is an iron center. They call it a heme iron center. So this is a compound, heme, and in the center of it is iron. Okay, so you can see here's the enzyme. So this is the protein. There's a heme molecule, and then there is this iron sitting in the center. Also attached here is something called an NADB enzyme, but don't worry about that for the moment. It'll make a bit more sense in a second. So what happens is this metal center has the ability to generate something called a hydroxyl group. Now, for those of you who are thinking now a little bit, or thinking, okay, this is too much for me, this is not essential to understand. I'm just answering Patricia's question. So if you're kind of finding it hard to follow this, don't worry. I'll do a bit of a summary at the end. I'm just answering a question. No, Patricia, you don't have to apologize. So just on that, so what we have here is you have this metal ion in the center of the enzyme. And so the metal ion acts as a cofactor. So this is hopefully now giving you an example of how cofactors play and how they work within enzymes. So in our P450 enzymes within our body, the metal ion at the center, which in this case is iron, is the cofactor. So it's essential for this enzyme to occur or to act or to work. And one of the things that it does is it generates a hydroxyl radical. So a hydroxyl radical is an OH with a little oxygen. So remember when we looked at the functional groups, the hydroxyl is a group and it has an OH attached to it. So autooxidation is the spontaneous oxidation of a substance in the presence of oxygen. So what oxidation is oxidation is the loss of electrons. So what autooxidation means here is a substance is losing electrons in the presence of oxygen. Reduction is the gain of electrons. So if you kind of even think back to just their energy production, they talk about the oxidation of glucose. And so glucose is losing its electrons, and they're then getting fed into the electron transport chain. So Glucose oxidation is the loss of electrons from glucose, and those electrons then are fed into the electron transport chain and converted into energy. So with metal ions then, they have the ability to lose electrons. And there's something really special in a sense about iron in particular. There's a couple of different ones, but iron has the ability to lose 2 or 4 electrons. It's a little bit different, or 3. So if I show you here. Okay. So we talk about this thing called valency, okay? So the valency of an element, sorry, I created this today, so I'm the worst proofer. The valency, and just stay with me here because hopefully this will make sense in a second. The valency of an electron is the number of hydrogen atoms that it can combine with, okay? And it's dependent on the number of electrons in the outermost shell. So all the valency means is that, so if we take oxygen has 2 electrons in its or 6 electrons in its outermost shell, so it can bond with 2 other molecules. Carbon has 4 electrons in its outermost shell. It needs 4 electrons. So we call that, say that it has a valency of 4. So most of the time, atoms or elements have one specific valency. So oxygen will most of the time have a valency of 2, carbon will have a valency of 4. But some metal ions have multiple valencies. So iron is one of those, and that's what kind of makes iron special, and it's actually what makes iron has the ability to transport oxygen around the body. So the iron ion, so it comes in 2 different forms. So sometimes iron can lose 2 electrons to form Fe2+, while other times it can lose 3 electrons to form Fe3+. So it has this ability to have multiple valencies, that's what we call it. If you don't get the valency thing, don't worry. Just take here that iron can either lose 2 electrons to become 2+, or it can lose 3 electrons to become a 3+ ion. And the reason it can do this is because it has a special shell. That, it has this special ability. Magnesium doesn't have this ability. So magnesium will always be magnesium 2+, and sodium will always be Na+, the sodium ion. But iron can sometimes be 2+ or 3+. And this is really important, and it's why hemoglobin actually works, and you can transport oxygen around the body. But so what happens here is, when we talk about metal auto-oxidation, Patricia, what we're talking about is the ability of the metal ion to release another electron. So it can change from Fe2+ to an Fe3+. So that's what that means. And that, sometimes we don't want that to happen, and sometimes it might be bonded to an amino acid because it can be so reactive. But sometimes that's actually a positive thing, that it can go from a 2+ to a 3+ affects its ability to act as a cofactor. So if we take this here, this is our cytochrome P450, and in the center you have this iron here, okay, and it's been held by these, it's not a covalent bond, it's a special thing. This is an iron ion in the center, and it can change between a 2+ and a 3+. And what that does is, because of that ability, it enables it to act as a cofactor. It's this ability to undergo auto-oxidation, to go from 2+ to a 3+ and lose an electron. So remember, that's what auto-oxidation is. It's the losing electrons in the presence of oxygen. So iron can go from 2+ to 3+. It can lose an electron in the presence of oxygen. And so this is how the CYP450 enzyme actually works. What happens is I'll show you here. This is our Fe2+ here. And what happens with this Fe2+, in the presence of oxygen, it goes from an Fe2 to an Fe3. So it loses an electron. And as a result, what happens is it it combines this hydrogen peroxide, and you generate this hydroxyl radical here. So this is a hydroxyl group, and it has an electron attached to it. And this guy, it's incredibly reactive. And what happens is, you learn about this in Unit 7, when you have compounds that need to be detoxified, and they go through 2 phases. They go through phase 1 and phase 2. And actually the detoxification process in the body, the first stage of it actually generates free radicals. Because what happens is our CYP450 enzyme has this iron center. In the presence of oxygen, it actually generates a hydroxyl radical. And that hydroxyl radical attaches to the compound that needs to be detoxified. So if we were to take, let's say, estrogen, I think it goes through this process. A lot of pesticides, actually, they have to, a hydroxyl radical has to attach to them. So all of the first phases, the first phase of if we jump back here, the first phase of detoxification, the CYP450 phase that sits on your endoplasmic reticulum or your mitochondria, it actually generates free radicals, and it makes the compounds that bond to it, the free radicals, more reactive, and that actually then enables them to go through a second phase. And that's why you learn about this a lot in Unit 7. Detoxification in the body is inherently potentially damaging if the second phase isn't working properly. Because if the first phase we're overreactive, it generates free radicals. And if the second phase isn't there to mop up the free radicals and to mop up the reactive products that are produced, it can actually create more damage. And we call this pathological detoxification. It's genuinely a problem that happens with people. And then just on Dermot, on your point there, is the enzymes for phase 2 are really, really, really important. And if, for example, you have SNPs or you break errors in the genes that code for those enzymes, people have issues, and you can do different things to kind of support them. But that is how actually detoxification itself is quite damaging. Not damaging, it's potentially damaging if you don't have these enzymes to mop it up. So Patricia, does that make kind of more sense, or does it bring that to life a bit for you more? Yes, much more. Okay, do you have any, hopefully that makes sense. Okay. Just to summarize for those of you who might have gotten a bit lost there and think, oh my God, why? Why is she telling me about metal ions and auto-oxidation? From this slide, it's just important to take that natural processes in the body are inherently reactive oxygen species generating processes. So energy production generates reactive oxygen species. Detoxification generates reactive oxygen species. And so does the breaking down of compounds, but also the immune system. And so it's really important to have protective molecules to negate the effects of these compounds. So if we jump on here, this is kind of a good picture that summarizes all of the content in this PowerPoint in a sense. So you produce pro-oxidants or reactive oxygen species in the body. So we've looked at where you make them. You also get them externally, so from UV light, X-rays, air pollution, chemicals, smoking, different things. And then we hopefully have antioxidants in the body that help to mop up or basically kind of create bonds with these compounds so that they can't do damage. So oxidative stress is where you have more pro-oxidants or reactive oxygen species than you do antioxidants. And as a result, you've more reactive oxygen species going around. And what they might do is they might go in and damage DNA strands. So remember, we have our lovely bases that are meant to be paired together very nicely. Sometimes reactive oxygen species can damage the bases and cause mutations. Or what can happen is reactive oxygen species can go into cell membranes, so our phospholipid bilayer here, and they can damage the fatty acid part. Because remember, particularly the unsaturated fatty acids where you have double bonds, they are really susceptible to oxidative damage. So if you have a lot of oxidative stress and you have a lot of reactive oxygen species, they can damage the cell membrane. And what happens is you damage the cell membrane or the immune system thinks that it's damaged, and it can then start to build up plaque, and it can damage the blood vessels. And what can also happen is reactive oxygen species can damage proteins or amino acids within proteins, and it can contribute to autoimmune diseases, because you basically make the molecules all of a sudden not look like part of the body, and the immune system thinks that they are foreign, so it attacks parts of the body. And also glycation of products. So reactive oxygen species can damage proteins, and they can then damage tissue as well. So that is kind of an overview. Then we just have here, so another question is, and my last question is related to antioxidants that can also be pro-oxidant. So far I can see that iron, vitamin E, and polyphenols, beta carotenes, they can be pro-oxidants, they can. Also, how do we know if they are acting as a pro-oxidant? So that is a good question. So we can look at this now. So just before we do that, just to look at, so antioxidants. So antioxidants are compounds that can donate an electron to a free radical, thereby stabilizing them and preventing them from destabilizing other molecules. So that's what antioxidants do. They give an electron to a free radical. But a lot of the time, in giving an electron to a free radical, they end up then becoming pro-oxidant themselves. So in relation to your question here, Patricia, so many compounds in the body, it's hugely broad, have the potential to be antioxidants and sometimes pro-oxidants. And what's really meant to happen is that the compounds in the body are meant to work together, and that's why the antioxidant network is so important. So if we look at antioxidants, there's lots of different categories of them. So you have, the biggest thing to look at is the two categories. So you've got antioxidant enzymes and non-antioxidant enzymes. Antioxidant enzymes tend to be our first line of defense. So you have SOD, catalase, and glutathione peroxidase. So these are proteins, they're enzymes, and they have an incredibly important role. You'll find them in the mitochondria, where they kind of try to mop up the free radicals produced by energy production. They are really important for detoxification. So they, again, negate the negative effects of phase 1. And then you find them in lots of other different places in the body. So they're really important. And these enzymes they have cofactors in the center of them. So they have, for example, SOD has zinc, copper and manganese. Catalase has iron, and glutathione peroxidase has selenium. So all of these minerals are really important for these enzymes to function properly. And so you'll see that one of your questions in assessment 2, question 5, asks you, what are the cofactors in those enzymes? Then you have non-antioxidant enzymes. So when we're looking at this, we're really looking at, there's a couple of different things. So we're looking at vitamins. So vitamin A, C, E, and K, all of them can act as antioxidants. And we've antioxidant cofactors or compounds. So CoQ10 is an antioxidant. And then we also have proteins. So organosulfur compounds. So lipoic acid is a compound, but so is glutathione. So glutathione is a really important protein. It's made up of cysteine, glutamine, and glycine. It's made up of 3 amino acids, and it's really important it acts as an incredibly important antioxidant. It's also really important in liver detoxification. So they're kind of some compounds. You've got proteins, vitamins, cofactors, and then you also have phytonutrients. So we won't have time to go through them all on this tutorial, but you've got lots of different phytonutrients in food, mainly in colorful food. So you've got the carotenoids, you've got beta carotene, lutein, xanthine, and you've got polyphenols. So they include flavonoids, phenolic acids, and they're a huge family. And you have other ones as well. And all of these phytonutrients that are found a lot of the time in plants have the capacity to act as antioxidants. They also have, though, some of them have a lot of double bonds in their structure, and that's why they're antioxidants. But that can also lead them to becoming pro-oxidant if they are in a highly reactive environment. And to be honest with you, we don't know the answer to that your question, really, in a sense, Patricia. Like, when does a carotenoid become a pro-oxidant? And we're not always sure. It's usually what happens is in nature, ideally, you have all of these compounds working together. So, for example, if we take vitamin E, if you give really high dosages of vitamin E, it can act as a pro-oxidant because if there's not other nutrients to kind of mop up the effects of it. So if I show you here, so this is vitamin E. So vitamin E, where it acts as an antioxidant is it lets this hydrogen on this ring, it goes off and it bonds with the free radical. But in the process, what you're then left with is an unpaired electron on the vitamin E. So an oxidized vitamin E is a pro-oxidant. It is potentially going to react. But usually what happens is naturally vitamin C will bond with vitamin E, donate a hydrogen to it, and then it gets regenerated. So what happens is it's really important sometimes not to give really, really, really high dosages, or to be cognizant if you're giving high dosages of vitamin E, it's important that there's vitamin C in the system as well, because they should balance each other out. And that's how the antioxidant network works. So if we take this here, and this again relates to one of your questions, how does the antioxidant network work? So the antioxidant network is a number of different compounds of antioxidants that work together to regenerate or recycle each other. So you can see here, free radicals are generated. Sometimes carotenoids can generate free radicals. And the free radicals here, if they react with vitamin E, they are neutralized. Okay, so they're not reactive anymore. But what happens is vitamin E in the process, it becomes more reactive. So in order for it to be neutralized, it reacts with vitamin C, which then vitamin C in itself becomes reactive. It reacts with the thiol cycle. The thiol cycle is usually made up of glutathione. So glutathione is a protein that works with vitamin C and it regenerates it. And then glutathione itself reacts with NAD, and then it in itself becomes regenerated. So all of these antioxidants work together to recycle and regenerate each other so that they can then be continuously used as antioxidants. And so what can happen is you need to be careful not to give really high dosages of certain nutrients. It's better to give an antioxidant in combination with each other so that it's that they are working synergistically. So there could be questions like, sometimes with, let's say, phytonutrients, and if you're delivering really high dosages of phytonutrient extracts, it's just important to be cognizant that sometimes in very high dosages is there the potential for them to become pro-oxidant, and it's probably better to be eating whole foods where there are not other antioxidants within it that will kind of balance each other out. And we don't fully understand how all of that works, if I'm honest with you, because not always the research is done. So that's the antioxidant network. It's made up of vitamin E, C, coenzyme Q10, CoQ10, glutathione, lipoic acid, and these compounds work together to regenerate each other so that they can continue to act as antioxidants. There's another picture. This one, the thiol cycle, it's referring to glutathione, but lipoic acid can be in there as well. Glutathione is thiols are things that have sulfur within them. So glutathione and what was the other one? Sorry. Lipoic acid. Sorry, yeah, lipoic acid. They have sulfur in them, so they're part of that cycle. But all you're really explaining here in the assessment is that these are the different nutrients that work together to regenerate each other so they continue to act as antioxidants. So Patricia, does that make sense, or do you have any more questions? Okay. So then the last part here is, so I can see on page 54 that flavonoids could have a cytotoxic action. Does that mean that they are pro-oxidants, or what is the toxic action they might have in cells? So what Patricia is referring to here is, so these are all polyphenols. Flavonoids are parts of them. And so this is the slide that Patricia is referring to. So it's overall as a group, they can generally have actions that include antioxidant, antimicrobial, cytotoxic, anti-inflammatory, and antiviral. So these phytonutrients, they have the potential to have multiple different effects within the body. And this here is, they're saying they can be cytotoxic. So when we are talking about cytotoxic, so cytotoxic can have lots of different meanings in different contexts. So the cytotoxic itself means it's the quality of being toxic to cells. A lot of the time when you will see the word cytotoxicity in journals or articles or books, a lot of the time they're looking at the side of its ability to have an anti-cancer effect, that it can be toxic to cancer cells and kill it. However, you might also see cytotoxic, like a certain compound can be cytotoxic, a toxin can be or a compound can be toxic, i.e. cytotoxic at certain levels. In this context here, it's referring to the article is talking about the potential of flavonoids to have an anti-tumor, anti-cancer effect. So some phytonutrients are thought to exhibit anti-cancer effects through the induction of cell apoptosis. So they can basically induce a process within the cell. Well, it's thought that they may be able to induce a process within a cell called cell apoptosis. And cell apoptosis is the death of cells, which occurs as a normal and controlled part of an organism's growth and development. So all of our cells in the body have the potential to undergo cell apoptosis, so cell death. And it's an organized, controlled process that it's almost like a protective mechanism, that if the cell becomes damaged or if the cell is getting worn out, that the cell has the ability to kind of to die in a sense. And so what they think how certain phytonutrients might have anti-cancer effects is that they can induce cell apoptosis in a cancer cell. So usually the cell should be able to spot themselves if they become cancerous and they should kill themselves. But if they don't, some researchers think that certain phytonutrients like flavonoids may be able to induce an anti-cancer effect through inducing cell apoptosis. And they do this through affecting, it's really, really complicated. They do it through affecting cell signaling and basically affecting different stages of a cell cycle. So cells go through this phase of growing and splitting of DNA and then splitting themselves naturally. There's this thing here. Again, you don't need to know this, but this is the cell cycle. They go through this thing called growth, DNA synthesis, growth and repair, and then they kind of split. And so how they think that these flavonoids work or have the potential to work is that they can basically induce cell apoptosis by affecting different phases of the cell cycle and killing certain cells. But again, this is by no means suggesting that one would use flavonoids as an as a treatment of cancer, but maybe it might prevent early stages of any kind of cell damage or something. Yeah, that's a good point. Yeah. So that is kind of one of the ways in which flavonoids might work. So that is kind of an overview of antioxidants. As I said, you will come to this again if, for those of you who are just want to see what is the key take-home points, I would say just having an awareness of what free radicals are and that they are pro-oxidants or reactive oxygen species. You don't need to know all these guys, but just have an awareness that you can make them inside the body and you can make them through energy production, the immune system, and detoxification. You also can generate them from exogenous sources, and collectively they will lead to a buildup of reactive oxygen species in the body. Ideally, what happens is that we have antioxidants to negate the effects of that. So we have antioxidant enzymes, antioxidant vitamins, proteins like glutathione and lipoic acid, and then also flavonoids as well, or phytonutrients, I should say. But if you don't have a good balance between pro-oxidants and antioxidants, you can have a level of oxidative stress in the body. And that in itself then can damage DNA, cell membranes, amino acids, and different compounds. So I said you'll come back to this when you touch on it in Unit 7 when you're looking at how to address biotransformation imbalances next year. You'll also look at antioxidant sources in Unit 8 as well when you're looking at how to address energy imbalances. So then finally, so this is really short. I just want to show you this pathway. So some of you might have heard about this before. So the methylation cycle is a number of chemical reactions in the body that generate a methyl group, and it's really important for a number of different reasons. And you will touch on this. This will come in to lots of different places. You'll learn about methylation when you're looking at the immune system. You'll touch on methylation in particular when you're looking at detoxification, and when you're looking at hormone balance, it'll come up again and again. Transsulfuration is another pathway in the body that generates glutathione and other sulfur compounds. What we'll just do now is look at these two pathways. So this is them here. I've just got one slide that summarizes it. And this is also your assessment question 7. So where will we start? So let me just show, this is kind of an overview. This is the methylation cycle here. It's kind of this part. And this is the transsulfuration pathway. So they're interlinked. What links them is actually homocysteine. So some of you might be familiar with homocysteine. It's a compound in the body, and it's thought to exhibit kind of inflammatory effects. So they think that a buildup of homocysteine isn't healthy, but it can also be a sign that your methylation pathway cycle is not functioning optimally. or your transsulfuration pathway. So what I'm going to do now is kind of show you this pathway. So if we start with protein, so when you eat protein, one of the amino acids in protein is methionine. And ideally, methionine gets broken down in the body. It'll get used for different things, but one of the things that it's used for is to generate methyl groups. So methionine gets converted into SAM, or S-adenyl methionine. I think that's the word. But anyway, it gets converted into SAM, and SAM then gets converted into SAH. And this process here generates methyl groups, and that's really, really, really important, because methylation, or the adding of a methyl group to lots of different things has a really important role. So it's an important role in neurotransmitter production, in estrogen metabolism, in detoxification, in regulating gene expression. So methyl groups have a really important role in the body. And so it's really important that this pathway is functioning. So ideally, methionine gets converted into SAM. This methyl group is released when you convert SAM into SAH, and then SAH gets converted into homocysteine. So homocysteine, it can then get converted into go this way, or it can get recycled here. And this is one of the pathways. So what happens is homocysteine gets converted back into methionine. But this process is really dependent on an enzyme called methionine synthase. This guy here, MS. And methionine synthase needs vitamin B12. You can't really see this that well, but this is vitamin B12. Methionine synthase needs vitamin B12 to function optimally. And so if you're low in vitamin B12, it can affect this pathway of regenerating methionine from homocysteine and ultimately creating methyl groups. So methionine synthase is important, and so is B12. So what also happens here is this is another cycle. It's called a folate cycle, and it supports this pathway too. And so what happens is folate, if we jump back here for a second, folate here is what you find in food. It's called dihydrofolate. Now you can get synthetic folic acid in a supplement, but ultimately that will get converted into dihydrofolate in the body, and dihydrofolate is what we need. So dihydrofolate gets, I'll show you here. Dihydrofolate then gets converted into tetrahydrofolate. So we have here, folate gets converted into tetrahydrofolate, first di, then tetra, and this tetrahydrofolate then gets converted into 5,10-methylene tetrahydrofolate, and then 5-methyl tetrahydrofolate. And so the production of methyl tetrahydrofolate from this one, methylene tetrahydrofolate, is really dependent on this compound, or it's not a compound, it's an enzyme called methyl tetrahydrofolate reductase. There's the or at the end. So methyl tetrahydrofolate reductase is a really important enzyme that enables you to generate methyl tetrahydrofolate. And this is also a really important compound because it's also carrying a methyl group. Okay, so you really want to be able to make methyl tetrahydrofolate because it plays an important role in this cycle here as well. And if, for whatever reason, you have a SNP in the gene that codes for methyl tetrahydrofolate reductase, you can have issues with making methyl tetrahydrofolate. And that's why sometimes you can buy in supplements methyl tetrahydrofolate. So it can, this is the active form of folic acid, they say. And so methyl tetrahydrofolate combines with homocysteine to make methionine. It's dependent on the enzyme methionine synthase, which also needs B12 as a cofactor. And so this is the pathway. So you might have heard about this before, or you might have heard of people saying, oh, you need to supplement with B12 and folic acid, and betaine, because betaine can also play a role. We won't look at that this evening because I don't want to confuse you. But these are important nutrients to support methylation and the breaking down of homocysteine. And this is basically the pathway and how it works. So I'll just talk you through it once more. In terms of the assessment, you just have to know what methionine is. This is B12, methyl tetrahydrofolate reductase. You need to know these names. So methionine gets converted into SAM, which gets converted into SAH, but in that process you release a methyl group. The SAH then gets converted into homocysteine. Ideally, homocysteine goes back up to methionine, but it needs 5-methyl tetrahydrofolate to do that, and the enzyme methionine synthase, as well as B12. You make methyl tetrahydrofolate from tetrahydrofolate, which comes from folic acid, but you are dependent on the enzyme methyl tetrahydrofolate reductase to do that. And if you have a SNP in that enzyme, sometimes you can have an issue with making methyl tetrahydrofolate. It's a bit of a tongue twister. And so that's why you will hear, if you go to conferences and different things, they'll often bring up the methylation cycle and people who have SNPs in the enzyme that codes, in the gene that codes for methyl tetrahydrofolate reductase. And so there are different things you can do to work around that. So that's the methylation cycle, but what people talk about less is actually the transsulfuration pathway. And so this is this pathway here. And what happens here is homocysteine gets converted into something called cystathione in the presence of the enzyme called CBS. I can't remember the exact name of that enzyme, but it's called CBS for short, and vitamin B6. The key thing here is that vitamin B6 is the cofactor for that enzyme. So converting homocysteine into eventually cysteine here is really dependent on vitamin B6. And we really want to make cysteine for a number of reasons. So cysteine is an amino acid. You can get it from food, but you also, as I said, make it from homocysteine. And it's an amino acid that has a sulfur group within it. And cysteine can get converted into a number of different things. It can get converted into another amino acid called taurine. And that's important for lots of different things. It's important for biotransformation. It's also important for neurotransmitter production. It helps to make GABA. But cysteine is also then important for making a protein called glutathione. And by the end of in 2 years, you will definitely know what glutathione is. You'll come up again and again. It's a really important protein. It acts as an antioxidant. So it's an incredibly important antioxidant in the body. It's also involved in detoxification. It's a really important role in detoxification, and you'll learn about that in Unit 7. And glutathione, it's made out of cysteine, another amino acid called glutamine, and glycine. And it is, though, highly dependent on cysteine. And sometimes people, they don't buy, if they want to supplement with glutathione, instead of actually buying glutathione, they buy cysteine, N-acetylcysteine, and that helps to build up levels of glutathione in the body. So it's a really important amino acid. It can also get converted into sulfate, which is a source of sulfur. Sulfate is really important in detoxification, as well as involved in protein synthesis as well. But the key one that you'll learn about is sulfate is a really important role in detoxification, breaking down compounds. So that is the two pathways. You've got the methylation pathway here, and this folate cycle here. Don't worry about this one. We'll come to this again. For the moment, if you can just take in that, and even just tetrahydrofolate goes to methyl tetrahydrofolate in the presence of MTHFR, that's a good start. And then this transsulfuration pathway helps to make sulfur compounds. That's why it's called sulfuration. And the key ones is taurine, glutathione, and sulfate. And again, you'll come back to these again and again. When you look at, when we go into detoxification in Unit 7, you will go through this pathway again, because these compounds are really important for detoxification. So that is the two pathways. This is just explaining, this is, I just put this in because I want to show you how important cysteine is. So it can be, cysteine can be used for protein synthesis, glutathione production, taurine, and sulfate. And the production of cysteine from homocysteine requires B6. And this is the enzyme here, cystathionine B synthase, or CBS. So that is there are the two pathways. As I said, don't worry, you're going to come again, you're going to see these again and again. This is just an introduction to them. You will see them again and again. So then just finally, I want to show you. So this is your Unit 2 Assessment 2 set of questions. We've looked at some of them last week. These are the ones that we touched on this week. So describing the role of a coenzyme and where they are, and what they're derived from. We looked at that this evening. List the cofactors for the link reaction and the coenzymes and the electron transport chain. We looked at that this evening. List the cofactors in antioxidant enzymes. We touched on that as well. Briefly describe how the antioxidant, what is meant by the term antioxidant network and how it works, let's say. Which well-known group of phytonutrients is known to exhibit both an antioxidant and pro-oxidant effect. So we looked at that, the carotenoids. This is the diagram that we just looked at here for question 7. And then this other question here, question 6, is define the term nutrigenomics and briefly outline its role in personalized nutrition. Outline the current strengths and weaknesses of personalized nutrition in terms of the strengths and weaknesses of, in a sense, the field of the research. So all of that you can find in this article here. So if you read this article, it explains what nutrigenetics and genomics is. And then it also gives you a summary of the strengths and weaknesses of the research here. So we just want to see that you've read the paper and that you have an idea of what nutrigenomics is. You just have to define the term. You can take that directly from the article and then just outline the strengths, the weaknesses, and you'll find that within the article itself as well. Okay. So does anyone have any questions on the assessment or anything? Will we need to reference? No, you don't need to reference. Not for this one. If you choose to include a paper, so we're not asking you to, let's say here, reference, because we're just asking you to read that paper and just summarize what's in it. If you have read another paper and you want to cite it, then yes, and you want to refer to it in your answer, definitely you can then cite it. But if you just include that paper, it's fine because we've asked you to read it. Yeah, so no, you can just write, let's say, number 1, 2, 3, and 4 and what they are beside it. So Deirdre, just say, so there's a PDF reference in question 5. Do we reference that? No, you don't have to. So just in terms of, I know this is for some people the first time that they've ever studied chemistry, and it's definitely, the 4 weeks, there's a lot of content in it. So I just encourage you, I know I've said this a number of times, but I just want to reiterate it, is that you don't need to leave this unit knowing everything. If you have an overview of a general understanding of the different concepts, that's a really good start. You're going to come back to them again and again. Next week, there is a reading week. So we put that reading week in to give you space between this kind of new heavy information and Unit 3. So unless you are incredibly motivated and want to study next week, I would, just say take the week off and have a bit of a break, because again, you're going to come back in November, and you'll start with Unit 3. I'm the unit leader for that as well, and Unit 4, and it will look at the communication systems. We'll look then at the assimilation systems. So it's good to take the space next week if you can. So there's no specific reading? No, Catherine, there's not. There's only, Catherine, so there's no specific reading for the reading week? No. Like, to be honest with you, the reading week should probably be called having a break week. If you want to read through the content to kind of ground yourself in it, great, but there's no additional reading for you, it's there to give you a bit of space to process what you've learned. Kids are off their break, it is, yeah. Okay, great. And then just finally, Patricia, in relation to your question earlier, so Unit 2 is open until Sunday. So if you have any questions on the content this week, or as you're, let's say, answering the questions, if questions might come up in relation to the content, the form is open and I will answer questions this week. But come Sunday, the unit will be closed. So I won't be answering questions on Unit 2 content after this week. Naturally some stuff might come up in the units that come up. Of course, you can ask questions then, but in general, the unit itself is actually closed. Okay, great. So if no one has any questions, I will leave it at that. Not at all. Okay, brilliant. So everyone have a lovely evening and good luck with the assessment.
Thanks, Maura. Bye-bye.
Thanks, Dermot.
Thank you. Thanks, Maura. Bye.
Bye.